Thank you all for showing up on I guess, Friday afternoon for many of you. So as Tom mentioned, I'm a physicist working on biology. And I thought it's maybe good to begin with, what draws us physicists to working on biology in the first place. And I feel the motivations can be quite diverse. Sometimes the attraction is something familiar. There's some biological phenomena, but then you see something in it, maybe a subset of it, that looks like a physics problem you solved before. But sometimes it's what's not familiar. It's examples in biology that you've never really thought about in physics. The focus of my group recently, and the focus of this talk, is more of the latter. A kind of phenomena in biology that you don't tend to think about in physics very much. And this is how we think about it. That in physics, you're often solving what are called forward problems. You take a bunch of particles with some fixed design parameters, maybe their interactions, the composition of some material, and you compute what the physical properties of that material are, band structure, transport properties, and so on. In some parts of physics, but especially in engineering, you often solve what's called the inverse problem. You know what properties you want, and you ask, what are the design parameters that will give you that? Okay. To us, the fascinating thing about biology is that it completely blurs the distinction between design parameters and the resulting properties, because the design parameters are often themselves dynamical and actively regulated by the current physical properties of the system. For example, if you're studying the swimming behavior of a bunch of bacteria, as a physicist, you may think of the flagella and the flagella motor, let's say, as a design parameter in your equation, as a fixed number. But in biology, when the bacteria swim for a while, and let's say their swimming isn't well adapted, there's adaptation and regulation which changes the structure of the motor, for example. Right? In fact, I think Robert Endres has worked on a lot of nice problems related to this phenomenon. And in fact, such regulation you find on a whole range of scales, all the way from gene regulation to evolution itself. Right? And further, even the nature of regulation itself can be regulated or evolved on longer time scales. More relevant for today, this kind of loop also describes learning in, say, neuroscience. Because your brain as it is today maybe has some synaptic connectivity. Let's call those the design parameters. That determines how your brain responds to stimuli as you walk around and listen to this talk today. But then, unfortunately, those stimuli will rewire your brain and change the design parameters, leaving you with a different neural network tomorrow, right? Hopefully a better one, but I don't know what this talk is going to do. And so this kind of loop is ubiquitous in biology, and it's not, and it really tells you that there isn't a notion of a fixed set of design parameters. Almost everything in biology is dynamical on some scale. And so in my group, we've been studying a couple of biological problems of this type. For example, recently we've started looking at the evolution itself, how evolution can select on things like mutation rates and change the speed of evolution. Or earlier we were thinking about circadian biology and how you can think of it as a dynamical system that changes its own behavior over time, its geometry over time. But again, relevant to today, we've been thinking about similar questions in synthetic contexts. For example, how do you create a soft material that's not designed to have some function, but a soft material that as you play with it physically, will physically change its own design parameters so as to get the function you want it to get, that it's physically learned the parameters needed to get where it should be. Right? And today I want to tell you about some preliminary work at the molecular scale that sort of starts down this line, just the start of this kind of research program. Right? All right. So let's begin with what is pattern recognition. And at this point, you've all probably heard more than you want to know about how neural networks are so amazing at classifying and detecting patterns. You could train a network with some images of cats and dogs, and it can recognize a future cat and say, oh, this is a cat. How? Presumably, it's learning some correlations pretty high order subtle correlations among the pixels that are common to all the cat images, some correlation feature that's common to the cat images, and using that to separate it from, say, the class of dog images, right? I want to ask a similar question in the molecular context. That is, can a soup of interacting molecules naturally show such behavior? Right? But what does such behavior mean? Well, it's not, the input is not going to be images of cats and dogs, but imagine the input is going to be a chemical context, a set of concentrations of many hundreds of species. So imagine your chemical environment is defined by the relative levels of, in this picture, 25 species. So let's say 100 different molecules, right? So in context one, maybe some subset of molecules are at high concentration and others are at low concentration. And that corresponds to cat1 or cat A, let's say. And a different set of molecular concentrations corresponds to cat B. And just like cats, no two cats are identical, these top two vectors. Can any of you see my, oops, can I point at something? Let me see. Can you see my laser pointer? Okay, great.
Yes, sir.
So, great. So, okay, so these two concentration vectors, right, might represent two chemical contexts. They may not be identical just the way no two cats are identical, but again, they may share some high dimensional correlation feature that's not shared by these concentration patterns that represent a dog or the class of dogs. And what I want to ask is, can a molecular soup somehow naturally classify any member of this class as class 1 by assembling some molecular structure in response, and classify these as structure as class 2 by assembling a different one, right? That's what it would mean to have a molecular neural network. And it would be especially great if that molecular soup could be, is trainable. That is, tomorrow, if, let's say, Tom wants me to add a third class of patterns, horses, I should be able to take the existing molecular soup, change it in some incremental manner, like train it further maybe, and add that third class to this, right? That's the hope. And I want to show you that essentially a set of promiscuously interacting molecules, self-assembling molecules with promiscuous interactions, naturally has this high-dimensional pattern recognition ability built into it, just because that's just the physics of self-assembly. The physics of nucleation and self-assembly naturally will do what's promised on the slide. Right. All right. Before I dive in, though, the people who actually did this work, which actually has been going on for some number of years, it got started really as a self-assembly problem with Zorana Zaravchik, who's now faculty at ESPCI in Paris. And then we wrote about the pattern recognition aspects with David Schwab in, again, 2 theoretical papers. And today I will also today I will focus partly on recent experiments led by Constantine Evans, who's a postdoc with Eric Winfrey at Caltech, and Jackson O'Brien, who is a graduate student here at Chicago. Right. So this last thing, there should be a preprint, hopefully soon. Right. Okay, so the broad outline is what? Can a molecular soup act like a neural network, right? Now that's a question that's been around for a while, and recently there's been experimental success. And I want to contrast what I want to show to this, because the approach here is quite different. The successful approach from Lulu Xian's lab at Caltech is what you could call a computer science approach. You take the neural network you want to mimic, and you translate elements of that neural network, neuron by neuron, into a set of chemical reactions. And so this pool of chemical reactions will implement element by element the neural network you wanted to mimic, right? And it's successful and it works well. What I'm going to tell you about, in fact, has a complementary set of strengths and weaknesses, but more importantly, conceptually, it's quite different. We're not going to mimic a neural network element by element. We're just going to look at the collective dynamics of many molecules, in particular the idea of crystal polymorphism, and it's going to mimic the collective dynamics of a neural network. And as I said, the central physics it's going to exploit is this idea of polymorphism, which you're all presumably familiar with. Like, I mean, you all know that molecules can form crystals, but actually for many molecules, there isn't just one crystalline form. There are many competing crystalline forms. Water has many competing forms of ice. And when you have such competing crystal forms, what happens when you crystallize this molecule is these different forms compete to nucleate. Different forms might have slightly different nucleation rates, and depending on how you cool your solution, you'll get more or less of one crystal form versus the other. Okay? This is a classic idea.
Arvind, can I just ask a question? Because I'm less familiar now with the neural type networks made from chemical reactions. So what are the requirements for these chemical reactions? I mean, just sort of anything non-linear, or what kind of chemical reactions are you talking about?
That's right. Well, so these are all implemented with DNA. I mean, the nonlinearity is the most important aspect because, right, that's what lets a neural network do a computation. In this paper, actually, I don't remember the detail, but you see there's the letters WTA in some sense. They stand for winner take all, which is obviously a highly nonlinear phenomena, and I forget how they implemented it, but the main reason they were successful here was they somehow managed to have this strong nonlinearity in these chemical reactions. But yes, in the past too, there have been some proposals, and the key element is always how do you get a strong nonlinearity in your chemical reactions? And that's actually a great question because sometimes maybe that's how I should have said this. What I'm going to show you is the nonlinearity is going to be nucleation. Yes, Rob.
Before they go, yeah, if you go back to that slide. Yeah, I mean, they've got the reactions on the right-hand side, right? The DNA is a way to implement an arbitrary A plus B goes to C plus D reaction. And on the right-hand side, they've got the reactions they use. And basically, the non-linearity winner takes all thing, if I remember, is because they have a fast annihilation reaction, which means that between all competing things, and then only the one that has the highest concentration gets left at the end to participate in another reaction through a much slower process.
Right.
So that's the third and the fourth steps in that thing there, yeah. But the DNA is just a way of implementing an arbitrary A plus B goes to C plus D.
In terms of the nonlinearity is the fact that chemical reactions, like 0 is a special number, you can't go negative, right? Is that maybe one way to say it?
Yeah, but I think as soon as you get bimolecularity, you get some kind of nonlinearity, and then it's a way. How do you make that as steep as possible?
As steep as possible. Exactly. That's right. Yeah. Robert, did you say something else, or?
No, thank you very much. Yeah.
All right. Sorry, I can't always see you folks. Like I think I can see the loudest, like the person who just spoke, but when you stop speaking, it switches to someone else. So I can't.
I think there's a difference between running the application and running it in a browser, but it's not really important.
All right. So So this was great because you sort of preempted the main theme, which is you need a nonlinearity. It's going to be nucleation, and this idea of crystal polymorphism is a classic old thing. We're going to use a twisted version of it, which is essentially crystal polymorphism taken over to biology. Classical crystals, you have 1 or 2 building blocks, and you build a large crystal out of it, right? Like sodium chloride has 2 building blocks, you build a giant crystal out of it. Imagine instead imagine a heterogeneous crystal where every building block is a distinct species, right? And the number of species scales with the size of the crystal you've built, right? Now, this is really only possible with biomolecules because you can generate an endless diversity of biomolecules through, let's say, sequence. But this right-hand side has so many new features that the left-hand side lacks. So, for example, the number of stable states, the number of phases, the number of crystalline forms on the left-hand side is almost always an order 1 number. It's just through some accidental features of this molecule, you may get 3 phases, you may get 9 phases. That's it. On the right-hand side, it turns out it's programmable, and it's an extensive number in the number of species, right? Like when you have n species, you can make your system have n programmable phases. And further, as I mentioned towards the end, in principle, these phases are learnable. Right.
So a quick question about heterogeneous crystal polymorphism. Is it proven experimentally? I mean, the fact that it can grow linearly with the number of inputs, I imagine there would be tons of metastable states that they can get stuck in and never result in the end product that you want.
Great question. So experimentally, in fact, the work I'm going to mention in a bit is the first that we know that has tried testing it, but it's not really a test of how far you can go. We put in 3 structures. Theoretically, you could simulate and see these obstacles to it that you mentioned, metastable states and problems. And at least theoretically, the simulations and theory where you see these metastable states, you can show that those problems happen after some number of programmed phases. Yeah, so the arguments are primarily theoretical. And as I'll mention later, similar questions have been thought about for neural networks. How many different sort of memories can you put into one neural network before they start interfering and creating metastable states? And there again, obviously the arguments are theoretical, but you know, there's a good combination of analytic theory and simulations supporting that. Experimentally, it's very much an open question.
Okay, thank you.
All right, these are great questions because it's really getting at the core themes of what I want to tell you about. And in fact, let me tell you the whole idea of this talk in about 2 slides with a cartoon, right? So this crystal polymorphism, let's think about it this way. Imagine your biomolecules that were going to form a crystal, think of them as pieces of a jigsaw puzzle set, right? And when you buy a nice, expensive jigsaw puzzle set, it turns out those pieces can fit together in exactly one way. Like the die cast they use to cut out the pieces, each shape is unique, even if it doesn't look like it. And they fit together in exactly one way to form a structure, right? You don't actually need to look at the image, it turns out, if you buy it from an expensive brand. Cheaper brands, that's not quite true. But now imagine that you carefully designed a jigsaw puzzle set with shaped pieces that could fit together in 2 completely different ways, right? In the red way or in the green way. And in the green way, pieces that were close together in the red structure are typically far apart. So pieces 1, 2, and 3 are close together in red and completely spread apart in the green, right? I want to emphasize this is the exact same jigsaw puzzle set of pieces. The color just shows you that they've been arranged differently, right? 2 globally different arrangements. And if you could do 2, maybe you could even do 3, right? Just imagine such a jigsaw puzzle set existed. So one set of biomolecules that can co-localize in different ways to form different structures. If you had that, I want to argue very briefly that you essentially have a molecular pattern recognizer because of the physics of nucleation. To show you that, let me stop drawing jigsaw puzzle pieces and draw them as square tiles that form 3 square structures, right? Same concept, one set of components that can arrange in one of 3 different ways to form structures. Imagine you had that. Now imagine that in your test tube, you had some molecules at a higher concentration than others, right? Just some random concentration pattern where some are high and some are low. Take that pattern and replot it on the spatial structure of these 3, right? Just replot on these 3 different structures. Now, those high concentration tiles, maybe they are scattered in the blue and the red structures, and maybe they happen to be co-localized on the green structure. Now, all 3 structures will have the same thermodynamic free energy because they have the same number of bonds, they have the same number of high concentration tiles, but the physics of nucleation will suggest that the central one will have a much higher nucleation rate and form to a much larger extent, self-assemble to a much larger extent than the ones on the right and left. Instead, if you took a different pattern, maybe the high tiles are localized on the red structure, right? It doesn't have to be perfectly localized. You'll get enhanced nucleation here and you'll get the red one. And if you think through that, it's immediately obvious that each structure has a natural set of patterns that it owns, concentration patterns that preferentially nucleate that structure. So one way to construct that, take the red structure, look at a little spot, right? Tiles that, species that are co-localized here. If you enhance those concentrations, those tiles would typically be in faraway spots in the green and in the blue structure. So this concentration pattern will preferentially nucleate the red structure over the green and the blue. If you are not getting this, please do ask a question, because this simple idea really, if I explain it right, it's a simple idea, is sort of at the heart of this talk. All right. And if you can do that, you can pick a different patch on the red structure, and it'll give you a different pattern that will again preferentially nucleate red, and so on for the green and the blue, right? And the way I'd like you to think about it is there is some high-dimensional abstract concentration space. This is a three-dimensional picture I got from Google Images, but imagine some high-dimensional space of concentration patterns, and that whole space is divided up into red, blue, and green regions separated by these separator surfaces. In each of these regions, you only nucleate one of these three structures, right? There'll be a set of patterns, some regions that prefer the red structure, some regions that prefer the blue, and so on. And there'll be decision boundaries between them, regions where the concentration pattern won't be so specific in who it nucleates. Yeah. We can be. There's a question.
Arvind.
Yeah.
Arvind, hi, Kashif.
Yes.
So I just have a quick question. So if I understood, I get the idea about the nucleation, but is one of the requirements that the self-assembly has to run through to completion? Because if that's so, then you might get some kind of depleted concentration in some of the patterns, which may not allow completion of assembly. So are you searching for complete assembly or partial assembly?
Great question. We're looking for complete assemblies because you want to have nucleation and then the full growth process. So the nucleation is sort of the non-linearity we are relying on. And I think what you said was, if you're looking for complete stuff, then depletion is going to be a problem. Is that
yes?
Right?
Yes,
so
exactly.
Great. Right. These are great questions, because it turns out in reality, there will be strong depletion effects, but it turns out the depletion effects work in our favor to enhance the nonlinearity that's inherent to nucleation. In that, depletion effects will sort of result in a winner-take-all effect, that if there's the pattern that, let's say, slightly prefers the red to the green and the blue, turns out depletion will just enhance that effect further and enhance the ratio of red to green to blue and make the results look even better than you might have expected without depletion. And I'll get to that in a moment.
That's interesting. Thank you.
Thank you.
Arvind, I've also a question, actually. I mean, I also understand the concept of it. I mean, you put these different patterns on your different grids, and on one grid, these molecular species are next to each other, and I guess then you say they have a good binding energy, and that would be a favorable condition. But you're talking about nucleation at the same time, and it seems to indicate something dynamic. So yes. I'm not fully understanding sort of the dynamic stuff of it, and how you would implement something like that dynamically, and then with a simple analogy of the grid somehow. It's
I think so. The second part of the statement I agree with. I didn't really say anything about binding energy. So the binding energy is exactly identical for all of these. So these are 25. In this picture, there are 25 species that are happy to sit together as in the red structure or in the green or in the blue. All the binding energies, let's say, are equal. There's no modulation of binding energies any further. It's all going to be about the second part of what you said, nucleation phenomena.
So I thought they have to be close together somehow on your grid.
Oh, good. Right. Meaning, so if you enhance the concentration of 5 components, and those 5 components happen to be close together, then I say that the nucleation rate, then I claim that the nucleation rate of red will be dramatically enhanced, whereas the nucleation rate of green will not be enhanced so much. Because what is the nucleation rate of green? In fact, that's sort of my next slide. But the claim is, nucleation cares about local enhancement of concentrations. It doesn't care about enhancement of concentration that's scattered. And hence, there's a preference. In fact, that's, in fact, just to answer Robert's question, so you can be a little more quantitative about this. So here is, on the left is classical nucleation theory. So classical nucleation theory is for homogeneous crystals. Imagine that simply saying that all concentrations are equal, right? What is nucleation really, for those of you who may not be familiar? All it's saying is that even if a crystal structure is favorable to form from monomers, the monomers first have to climb a hill before they have to go down, even when the structure is favorable to form. Why? Because if you compute free energy, it turns out there is an area contribution and a perimeter contribution that oppose each other, because the usual story, that's energy from the bonds, but there's entropy from translational losses. Bottom line, there is a critical seed size you need to assemble before things are easy and you grow out, right? Now, and you can compute the nucleation rate with Arrhenius's approximation and all that. But here's the key point. Imagine you have a crystal with unequal concentrations, okay? If you have a crystal with unequal concentrations scattered across the crystal structure, you can draw a similar free energy versus structure size picture. It's going to be a much more complicated graph. And if the concentration pattern, the high concentrations are scattered all over the structure, you'll get some graph like this with a high peak, because a critical seed might be a large one. But if those concentrations are close together, if the tiles with high concentrations happen to be close together, the free energy surface will look more like this with a lower barrier, because essentially it's saying the moment you assemble that little piece of high concentration tiles, everything is downhill from there, right? So the peak you have to climb before you go downhill can be much lower if you collect the concentrations. Does that make some sense, Robert? Or? I'm saying classical nucleation theory, if you take it over to uneven concentrations, you will immediately get a scale dependence, a spatial pattern dependence. Right? In fact, you can be more precise about it. It's about enhancement on a particular length scale, A. What is that length scale? It's set by temperature and other parameters. But here is a rough heuristic, right? Let's say this is the concentration pattern. These tiles are at high concentrations, the marked ones. Where will the structure nucleate? Well, you can probably intuitively guess, probably around here. The correct way to do it is you have a rough guess for the structure, the size A of the critical seed, and you look across the structure over regions of size A and ask, where do you find the highest set of concentrations? That's the place where things will nucleate, right? It's a rough heuristic. This is not reality, because that size A itself depends on concentrations, and you have to do a more complex thing to solve this. In fact, kinds of methods that Tom has in fact developed for sampling rare events. But let me give you one consequence of this to see if you get the intuition behind this. It turns out if you carry out these kinds of experiments at high temperatures, your experiment will be much slower, because nucleation is always slower at high temperatures. But you can solve more complex pattern recognition problems. You can separate cats from tigers. At low temperatures, the experiment will be faster, but you can only solve simple problems. You can separate cats from toasters, maybe. And here's why. This is your concentration pattern. And imagine it's localized extremely well in structure 1 and slightly localized in structure 2. If you carry out the experiment at high temperatures, you care about localization on a large scale. And on a large scale, green fails, red succeeds. The ratio of nucleation rates will be a large number, meaning you're sensitive to high order correlations. But if you do the experiment at low temperatures, maybe all you care about is localization of like a couple of tiles. And from that perspective, the red and green are equally good. The pattern is equally localized in both. And so you'll only be sensitive to low order correlations. You'll have equal nucleation rates. But on the other hand, it's a faster experiment. And in fact, you can do some theory to quantify a trade-off between time or speed and the complexity of the experiment, of the computational task you're solving. Like, what order correlation are you able to distinguish? This, right? So that's the theory of this. Does that make some sense? Or sorry. All right. That's making some sense. Let me show you briefly how would you make this. Right. So one way to do this is make a jigsaw puzzle set like this as well. You could cut out pieces of paper and make a jigsaw puzzle set. Or maybe there are proteins that have naturally evolved the interactions needed to assemble multiple structures. But another simple synthetic way to do this is with DNA, because with DNA you can design interactions pretty well. So one example from the Peng Yin lab at Harvard are these two-dimensional self-assembled structures. So this P right here is self-assembled from, I don't know, 300, 400 different particles. Each particle is just one strand of DNA, but each brick here, each little box you see here is a distinct sequence of DNA. And really, conceptually, it's very simple. Each strand effectively folds into a brick, exposing 4 sequences. And so on the computer, if you do your sequence design right, so that each binding site accepts exactly one partner, then there are 300 particles that like to fit together in exactly one way. So it's a usual jigsaw puzzle set from an expensive jigsaw puzzle set from a nice brand, right? And things just fit together and they assemble the structure. What we need is one promiscuous soup that can assemble multiple structures, right? Has the ability to potentially come together into 3 different things. And here's what we did. We did the simplest thing, which is actually inspired by the neural network analogy, which is we designed a set of molecules, in this case DNA, to simply have the interactions, to have all the interactions needed to assemble these 3 structures. We simply took the sum of the interactions needed. So if 3 needs to bind to 1 and 6 to assemble red, but then to 5 and 12 to assemble green, right, and to that and that to assemble blue, we just simply made each site, binding site of 3 promiscuous. That just added up all the interactions. And also in experiments, you don't want to make square structures. You want to be able to see what you get. So as a homage to Hopfield associative memory, which is a neural network context where you create a neural network with this kind of additive principle, we picked structures that look like this, right? And then you simply buy those DNA sequences, mix them together in one test tube, and I'm going to call that my master mix, my molecular neural network, right? For now, let's pretend we had 250 strands in this. There were some experimental complications or subtleties which actually meant we had some helper strands, but the conceptual story just think we had 250 such jigsaw puzzle pieces that could come together. And if this was not an online talk, I would go to my browser and show you a video from the Wyss Institute showing you how this actually happens in 2-3 dimensions. Right? Well, it's an animation, but still. Again, I mean, you can abstract it as a 2D picture for now. Okay, so here are the experimental results, right? So we have this master mix, and if you have a question, just ask, please, right away. It's just a mixture of these jigsaw puzzle pieces with equal concentrations for now, right? And just as a test of this, before we do any pattern recognition, you take this soup and simply cool it down on the cool it down, meaning you let things assemble. You make it favorable for things to assemble, and you look at it on an AFM, and you see this jigsaw puzzle set came together to mostly just make H's, A's, and M's, and you see many broken versions of it. But actually, you don't really see, at least not that you can resolve by eye, any unintentional structures that look like metastable states of something else. So like the earlier question, right, that it suggests that 3 structures you can put in and get exactly, you make such a jigsaw puzzle set and shake the box, you'll get exactly those 3, you'll get one of those 3 structures. All right, and you can zoom in and look at it. Right. So that's a neural network to which we gave no input, if you wish. But the real question is what happens if you take the neural network and enhance some concentrations, right? And we tried several kinds of patterns. Imagine you enhance concentrations in the left-hand side case that happen to be tiles that are all close together in H. So you see the little white pixels, they're meant to be high concentrations. So this picture, you should obviously expect it to nucleate H, because I've put in high concentrations that are close together in H, but they're scattered in A and M. Whereas here, they're close together in A, here they're close together in M. Right? And so you do the same thing. You just take the soup, enhance those concentrations, and cool it down. Right? Remember, the same soup, when you cooled it down a moment ago, it gave you H's, A's, and M's. But now with this enhancement, those particles dominantly form an H. That same soup with a different enhanced set of concentrations forms only an A. And the same soup forms an M, depending on the pattern that was enhanced. Right? So again, if you're not following the details, all I'm describing is this toy jigsaw puzzle set, but where we put in a few extra pieces of some of the some puzzle pieces and cool it down. So that's with an AFM. To be more quantitative, you can put in fluorophore quencher pairs that will quench if you assemble an H, but not an A or an M, right? Because the structures differ by co-localization. So you could put a fluorophore on one molecule, a quencher on another, that are brought together if you assemble H, for example. And when you do that, and we do that for all the three structures, and you can quantitatively watch it as a function of time. You see that the H signal quenches, but the A and the M don't really quench for a while. So all of these experiments are things in which we cool things down, right? So eventually everybody should really assemble things, but you see the H quenched a lot earlier and went much deeper. The others didn't as much. And in fact, in these examples, you see some non-specificity. The A, it's working. I mean, the A is nucleating first, or maybe growing first, but the others, which one is this? The M is nucleating to a bigger extent than maybe we saw in this one. Here it's pretty good again. All right, and okay, so that was one example. Really, we did a whole host of patterns, right? These are all different patterns. You could localized on H. These are all different patterns localized on A. Many different patterns localized on M, right? And then we put that all together into some summary statistics. The conclusion is it mostly works. When you localize on H, you tend to get H's, A's, and M's correspondingly. There are some failure cases, some of which are interesting. For example, if you look at the first pattern here, right, you see the pattern is localized on H, but just by accident, those high concentrations also happen to be localized on M, right? And so you could expect that this concentration pattern won't be as specific in H versus M, depending on the temperature you're working at. And we see some failures like that, and then also some failures that are unrelated that I won't get into. Right. All right, so we were actually surprised by how easily this worked when you just ramp things down, which gets back to one of the early questions someone asked, which has to do with depletion. So this problem of how, when you have a crystal with two forms, when you have a molecule that can form two crystalline forms, how do you get only one to form is a famous problem with a lot of industrial applications, because when you make drugs, you often want only one. Even more, to make chocolate. Chocolate, it turns out, has what? Five forms, and only the top form, the most stable form, this 92 Celsius. I don't know what it is. 92 Fahrenheit, probably. It's got to be 92 Fahrenheit. No, never mind. Who knows? Only the top form is a nice one that you want to sell, right? And so if you just take molten chocolate and cool it down too fast, for example, you'll get a bunch of the other types, which is terrible. And so what people have done, or people figured out more than 100 years ago, was a clever way to go up and down in temperature to form mostly the stable one, right? So you go up, melt, you come back down, you nucleate a whole bunch, but then you go back up to melt the bad ones without, hoping to keep the good stuff, and then you cool. And this is really a whole field in itself, how to get that. Folks with me so far? It's making some sense? But it turns out our problem is easier than this. So this is with classical crystals. In our case, the effects of depletion actually make the problem easier than in the classical case. And here's why. Imagine this is our temperature protocol, all right? We're just dropping temperature as a function of time. If our pattern is localized on H, let's say, when we get to this temperature, H will start nucleating. And then let's say as we go down in temperature further, those nuclei are going to quickly grow out. And as H grows out, it's using up components that A and M would need, right? And so, oops, so okay, that's H growing out. And because H has grown out, you've used up components, and the depletion essentially means that the nucleation temperature for A and M are going to drop due to depletion. On the other hand, the depletion temperature, the nucleation temperature for H is not going to drop because H is nucleating based on high concentration tiles. So that, let me just say that in a different way. When you assemble H and grow H out, the entire structure grows out, and all these tiles are used up. These low concentration tiles are used up, which are needed for nucleating something in A. But H itself is not affected by depletion as much because its nucleus is mostly made of high concentration tiles, which as a fraction barely deplete. As a result, you essentially, the winner, the one who gets to nucleate first, essentially grows, depletes, and kills the others without hurting itself, right? And you can ask whether this is true in our temporal data, and you can show that it's actually a pretty strong effect in our data, at least for most of our runs, right? It depends on exactly how you drop the temperature, but if you spend enough time in a region where H grows out fully, which I think exactly was the first question I got, you get to exploit depletion to make this a strong effect. And then we call that winner take all because, you know, essentially the first one who goes gets to win.
Hi, Arvind. Thanks for that explanation. That really helped actually. Just following up on that, then I guess that is dependent then on one having a sufficient concentration in order to have the grow out. So when you get into some kind of small copy number, then this may not occur, for example. Is that right?
Right. I
mean, or do you always find it?
Well, when we set up the experiment, we certainly have enough of these gray tiles to grow at least some H's, because otherwise, you know, why are you doing the self-assembly experiment without the components, right? But maybe you're asking towards the end of the experiment, how much is left? That we haven't thought through, but yeah, I mean.
Yeah, I mean, the reason for asking is because I think that in lots of, let's say, biological assembly cases, you might run into some kind of trapped states, like meta-assembled trapped states. And then how does a biological system overcome that? And that's kind of like, maybe this kind of process can help to explain that, but I'm not entirely sure.
No, you're right. That's right. So so these are synthetic experiments in a test tube with way more copies of molecules than you would have in a cell. If you had way fewer to start with in a cell, there are intrinsic problems to assembling a structure at very low concentrations that has nothing to do with what I'm talking about, right? That's just layered on top. But I think even if you only start with, let's say, 100 copies of each of these molecules, you still have a depletion-like effect. In fact, it might be much stronger. You only have 100 copies of these molecules. They have to choose whether to become H, A, or M. And if a few H's, if you form 50 nuclei for H, then game's up, right? The game's over for A and M, because all these nuclei are there, and they're going to use up half the products. Half the building blocks. But yeah, I mean, in that case, there is also stochasticity that we haven't thought about. But that's right. All right. Okay, so that's sort of the physics of it. But if you zoom out, sort of the conceptual question, what I've really shown you is sort of an obvious. I mean, once you understand the idea, it's kind of obvious that all I've done is taken H, enhance the concentration of molecules that I know are close together in H, not close together in the other two. And that gives me a pattern, and that obviously then nucleates H, right? And I did that for the others. The real question is, how powerful is this as a new sort of a recognizer, as a neural network like object. Meaning, if you give me patterns, right? You give me 3 arbitrary patterns, and you say these should assemble H, and whereas these should assemble A, and these should assemble M. Is this system flexible enough to do that? If you remember early on, I had this image I got from Google of a separator surface in high-dimensional space, right? How flexible is that surface? Can that separate, cats from minks, from tigers and lions? Or can it only do, is it a smoother surface that can't, do subtle correlations? That's really a question one should answer theoretically, first, I would guess. But we did a quick sort of experimental demonstration by doing the following, to just test how powerful this is. To test that, essentially, we need to pick arbitrary patterns. Yes. Yes?
You are muted, Tom. You're muted.
Sorry, how much higher do the enriched components need to be clearly detected like this? What's the fold difference?
I think the smallest we got it to work with, the experiments I showed you were pretty large, because it's a trade-off with time. I think the experiments we showed you might have been 20x. The smallest I think we ever got it to work was 2x. 2x or maybe 5x. It's just a trade-off with time and how. Yeah, it's essentially a trade-off with time, because you're dropping this temperature, right? And most of the temperature range, there's no action. Everything happens within a few degrees because it's melting temperature and all that. And so you want to spend enough time in that interval. So if you really know that interval well, and then you can dedicate all the time there, you could get away with a very small fold change. If you don't know and you just want to rush through, then you should have higher.
Okay.
A quick follow-up question on that. Would it be true then that the more clustered and differentiated you make them with respect to the other patterns, the lower you would expect this threshold to be?
Which threshold?
The threshold of concentration.
Yes. Right. Yes. Right. I mean, well, with the third node of the triangle being how long you're running the experiment. Yes. That's right. Right. That's right. Yeah, and in fact, that's pretty clear. Yeah, I mean, in the experiments, we fixed a standard protocol for everything, so we could run it on one machine and all that. But yes, that's right. But yeah, actually, in the early pilot experiments, we went too fast. Most things didn't work, but some worked really well because they were highly localized and others were not, and things like that. So, right. So to ask this question of how powerful is this, we wanted to challenge ourselves with arbitrary patterns. One way of doing that is to do a Google image search and treat images as concentration patterns. So these are images with so many pixels, the same number of pixels. This is an image of Dorothy Hodgkin. This is Avogadro. And this is Mitscherlich, who discovered crystal polymorphism. And Dorothy Hodgkin won the Nobel Prize for X-ray crystallography, at Avogadro. Well, all right. So these are 3 random patterns in some sense, right? We didn't get to engineer them. And we asked, can we make sure that this nucleates H, this nucleates A, this nucleates M? Except we already had these molecules. So essentially what we asked was, if you download such images, you still have a choice in how you name each pixel with the name of a molecule, right? Like you get to make this one choice once and for all the images together. Let's say this image has 900 pixels. We have 900 molecules, let's say. You get to make this one-to-one mapping between pixels and images once and for all. So after that, the pixel value, like how bright it is, tells you the concentration, right? And we asked, is there some assignment which will do this classification task correctly? And if I'm communicating this well, you should see that this is some way of asking, like, is this a flexible system? Right, that's a detail. All right, and so then we asked, and for these 3, it was actually pretty easy. You could, in fact, there were lots of choices where Hodgkin would, the white pixels in Hodgkin would be generally localized in H, but not in A and M, whereas Avogadro liked A. So then we pushed it further. We said, it's not just Hodgkin that should nucleate H. A picture of Hodgkin, John Hopfield, and a horse. These 3 unrelated patterns should all nucleate H, presumably by nucleating it in different places. Who knows? They should do that, while these 3 images should make an A, and these 3 should make an M. Right? This is Larry Abbott, a computational neuroscientist. This is Elizabeth Moser, who won the Nobel Prize for a kind of neural network, a real network in the brain that this system is closely related to. Anyway, so we asked ourselves this, and it turns out you could do this as well. And here are the experimental results. This is actually, right. So with these 3 patterns, this is sort of what we predicted would be the resulting concentration patterns. You do the experiment, and you mostly see quenching of H for all 3 patterns. You look at it on the AFM, you see H's. You try it with these 3 images, you get A's, right? With some, with variable specificity, right? Here you see, after a while, one of the wrong one does quench to some extent. And the same for M as well, right? Oh, and then this one actually, with M, you actually start to see one sign of a failure. And this is just with 3 images assigned to each category. You get this. And in fact, because it worked reasonably well for 3, we kept pushing it up to 6, right? That is 6 completely arbitrarily chosen patterns should all give an H, while these give an A and M and so on. And we,
sorry, trying to understand this. So it seems like if you are 30 by 30 grid per picture, then you just divide the total areas of M or H, then that's the amount of patterns you can have, right, roughly. Because you're just using a different areas of your H for different picture.
Except they should all work. So that map should work for everybody at the same time. So, right. So once and for all, we decide, before you look at any image, forget the image for a moment. We have this canvas, 30 by 30. Mm-hmm. What we get, the only freedom we have is deciding which pixel on this abstract canvas is which molecule in our experiment. That is, the grayscale value of that pixel sets the concentration of tile 27, whereas this pixel sets the concentration of tile 45. We decide that once and for all. And then we ask, is there an assignment that, will take the white pixels in Dorothy Hodgkin and localize it somewhere in H, take the white pixels of Hopfield and localize it maybe somewhere else in H, and the horse somewhere else in H, while also keeping them separate in A and M. And the same for Avogadro, Abbott, and this, right? Yeah. And so it's not obvious you can do that, because it's obvious that if I just asked you to get Hopfield's white pixels localized, you can easily do that in a million ways. But you also have the negative constraint of not localizing on the others, and then also the positive constraint and negative constraint for the other images. Right?
So each time you add a new set of images, you have to redo the assignment.
Yes, this was actually right, because this was actually a computer problem, right? So we did it on a computer, and this was a test of capacity. I wouldn't call this you could call it a learning thing, but it's not it's you're exactly right.
Yeah, you kind of did learning on the computer and then used the molecular system to display the results of the learning.
That's right. That's right. Yeah. This is not molecular learning. We're just asking what is the capacity of the system, so to speak. You do the learning on the computer. Yeah. When it starts failing, you saw some examples of failure, it's telling you that this is how flexible the system is as a classifier, like the 2-dimensional nature of these crystals, the nature of nucleation, doesn't allow arbitrary classification. You can't give me 20 images randomly chosen and say, make them all nucleate H, right? In fact, I think I took the slides out, but you can ask this question theoretically and you'll see that there's some capacity, like you can't just ask for an arbitrary number. We haven't yet figured out how it depends on the parameters, but yeah, I want to get the main intuition I'm hoping to get across is this is a test of how flexible it is, and you see that up to 6 it works to some extent. And in fact, the failures are actually quite insightful. It worked well for most of the images I'm showing here, but it doesn't work well for this column of images, these numbers, because as you can see from the numbers, actually, these MNIST digits, the white pixels are all pretty much in the same place for these 3, right? And presumably because of that, like this one had a spectacular failure. And when you have such a thing working, a natural thing to ask is, does it work even if you corrupt an image? Right. So you trained it with a nice horse, but then you give it a corrupted horse. Right. You just put some random pixels, and you try it. Naturally, it's pretty robust to this kind of corruption. Right? At least for this, it still works. But you can ask about a different kind of corruption, where you just obscure one band. And it turns out it's a lot less robust to this band. Wait, did I miss it? Yeah, well, at least you see, I think I had a more spectacular failure that I must have deleted at the last minute. But I guess the larger point here is anytime you have a classifier, a neural network or whatever, the architecture of the network decides what kinds of failures it's prone to and what kind of failures, what kind of distortions it's robust to. So if you have a convolutional neural network, you can translate images, and the convolutional network, will laugh at it. It has no problem with it. In the same vein, this nucleation-based system can deal with a lot of single-point mistakes. But if you obscure like an entire band, and that band happens to cover like all the bright pixels, it's pretty vulnerable to that, right? And certainly this is extremely vulnerable to translation. We didn't even try it, because if you translate it, the pixel to tile assignment would be completely confused. You would get nothing. And this is a quantification of all the data in some sense.
Just one question about the fluorescence data. I didn't fully understand how this is done. So you talked about sequencing. So let's say the red signal goes down. So what does it mean again? Because in the beginning, it looks like the red is actually passing all the other ones. And it seems like I predict already what happens later. I mean, it looks almost like you could basically already decide after a couple of hours, because A is somewhat dominating, and then later on it's quenched and you get the strong readout. But yeah. So what does it mean, the initial phase?
Yeah, I should have. So actually the rise is due to complications like I can tell you about, but it's really the fall. At least when we designed it, we were hoping to only see the fall. Because we put a fluorophore on one strand and a quencher on a different molecule. And so the signal should be bright if there's no self-assembly. When H is assembled, let's say the fluorophore and quencher were close together on H. When H is assembled, that pair should rapidly quench. Especially if that pair was in the critical nucleus, then it should dramatically drop, right? And not otherwise. And in fact, I didn't tell you the details, but essentially we put 4 pairs on each of these structures so we could monitor nucleation and growth. And we did the experiments with different sets of fluorophores present at the same time and so on. Okay, I guess I'm saying what we really have a good mental model for is the fall. The rise, it turns out, are the usual complications of fluorophores when you have other molecules around. Sometimes fluorophores get brighter if you just have other DNA around, for example. Right.
So it doesn't mean that the fact that red is high in the beginning, that it assembles less well in the beginning, and then all of a sudden later it assembles better. No, it doesn't not what it means.
No, we don't. No, we don't think that. And in fact, right, in these experiments, the temperature is dropping, so the x-axis is time, but the temperature is also dropping. So what this really means is in the early times, the temperature was just too high for anything to happen. And when you start seeing the drop, that's when it was cool enough for at least the H to start coming together.
And so these are different fluorophores basically. So red could be always higher because it doesn't really mean anything. The y-axis, so arbitrary units. Okay.
Well, we have normalized here, but right, in some way, right, the additional annoying complications of fluorophores, which make them a bit brighter or darker, can be different for different fluorophores, and that's why red is sometimes higher than right. Yeah, I would say the only robust feature here is a dramatic drop in one of the fluorophore levels. That's
I just want to also make sure basically there's no systematic bias, let's say, the red one, because it seems to be always the one which is winning. Maybe the red one just likes to assemble.
Yeah, great question. Good. So, well, on this slide, the red is literally the same fluorophore, but what do I want to say? So I've shown you many fluorophore traces, and when you look across them, that would answer the question, because essentially we picked 4 different dyes, right? Like I forget the names, but we picked 4 different dyes. One was rocks, for example, and I don't know if you can see this, but on H you see little colored pixels. And we use the 4 different dyes in those 4 different locations. And we use those same 4 dyes on A. And we use those same 4 dyes on M. Now, you can't do measurements if you're using the same fluorophores on different ones, but essentially we repeated the experiments in many, many replicates, where sometimes we would pick, this fluorophore on H, this on A, and this on M, and then repeat it with the fluorophores switched around to make sure the fluorophores aren't affecting, growth.
Yeah, sure. I mean, of course, here red is winning because red is the letter H, I guess, and
that's
all of these horses assemble effectively.
Exactly. But right. So I mean, I guess right. So here the blue ones drop first, whereas here the red ones, or here the yellow ones drop first, right? And in fact, yeah, here the yellow ones drop first, and so on. Yeah, all I'm trying to say is we switched. We have all the fluorophores and everything, and we have switched them around. So. Right. And this is sort of a summary of the data. You could try to quantify how well things worked, either by counting the number of images in an AFM image, which is that on the left. You could quantify the data with this fluorophore, like when things drop, right? This is sort of related to Robert's question. We look for a big drop. And you see that, and if it gets to the corners of the triangle, things are good. In the middle, it's not so great. All right. Any other questions? Because otherwise I'm going to wrap up with a little bit of theory at the end. All right. So this question was anticipated. Why do you just get these 3 structures? So in fact, naively, you should get all kinds of other metastable structures, because if you have promiscuous interactions, lots of other stuff should be possible in principle. We studied this question earlier and realized that in this system, when you have this promiscuity, right, when you program in 3 structures, you're making the system promiscuous. When you have a little bit of promiscuity, actually there turns out to be no problem at all. Wait, I had a graph. You have no problem at all in that these metastable states, you can show that their energies are actually way higher than you would naively expect, and that you don't really have many of them. But instead of 3, let's say you tried 5 or 10 or some number. At some point, this jigsaw puzzle set becomes so promiscuous, you get all kinds of combinatorial structures. There's a sudden explosion. And that happens at a specific point, right? It's obvious it should happen. What's not obvious and interesting is that it happens suddenly at some point. And it turns out it's related to a spin glass transition and related to why in a Hopfield neural network, you can store some number of distinct behaviors, memories, without getting them confused. But then after that, all hell breaks loose. But it turns out what I've been talking about is not like Hopfield's original associative memory, which has no geometry. It's in infinite dimensions. I have 2-dimensional structures here. And it turns out it's related to 2-dimensional neural networks of a kind that's been studied only recently, essentially because of this amazing work by the Mosers and O'Keefe on place cell networks in the hippocampus. I don't have time to go into this, but I'll just briefly mention it just because there might be Ising model aficionados. I know there are in the audience, and I think it's a really cool physics system that people haven't studied. So we all know what an Ising model is, right? And we know what a disordered Ising model is. You just add some random connections between them. It turns out for these place cell networks, those networks motivate a special kind of disorder. That is, red links like this, except red links that secretly form a distinct Ising model. That is, these red links look crazy when you draw them on the blue structure like this, on the Ising model, but there's some way to permute these spins, rearrange these spins in a different lattice, where the red links don't look disordered. It's actually another ordered Ising model, hidden, scrambled within this Ising model. And in the second perspective, the blue links look like disordered. Right? It's sort of 2 Ising models hidden inside each other, sort of like cryptographically hidden inside each other. And you can ask, what are the phases of that system? What are the phase transitions in that? And that question, well, you can ask usual questions about droplets and so on. Anyway, it turns out that answers questions about self-assembly as well. You can see this in our published papers, actually. All right. So actually, I'm bit over time, so I'm going to wrap this up. Now, this was, as someone pointed out, the learning in this happened on a computer, right? But our hope is someday we could have learning happen in situ, like molecular learning of some kind. The problem is, how do you learn interactions between molecules? Biology can teach interactions between molecules through evolution, right? Not in one generation, but through multiple generations. You mutate proteins and you can change things. Similarly with DNA, if you have sequences, once you buy those sequences, you have fixed interactions. But here's an idea. What if you chose your building blocks, I and J, to not have any direct interactions, but instead you mediated interactions between I and J through through a new molecule, IJ, right, which glues to either side. You could do the same with four-way interactions as well. You want IJKL to interact and glue together. Instead, you have a gluing particle, right, which mediates a direct interaction. If you have that, I'll explain in a slide, you could potentially learn interactions because these gluing particles might be created. You can create particles through enzymatic reactions, through ligation or other things. And so if you can create glue particles on the fly, you might be able to learn interactions on the fly. And in fact, the experiments I described, I kept abstracting it to the theory, but actually the experiments we described are not quite the way I presented it. What we really had was the set of shared tiles like I promised, but those shared tiles did not directly interact with each other. These jigsaw puzzle pieces didn't directly bind each other, right? These brown ones. Instead, we had one set of gluing tiles, red ones, that mediated interaction between the brown tiles to make them into an H, and a different set of green tiles that mediated this to make it into an A, and a different set to mediate it to make it into an F. Conceptually, it's the same thing, but physically it's different in that we have mediating particles, right? And we did this actually for an experimental constraint. And in fact, the real experiment, oops, something went wrong with the slide, but the real one was, deviated from this perfect checkerboard pattern in a few places to save money, but I won't get into the details. But we did this for a constraint reason. But at the end, we realized this could be to our benefit. Because if you have a gluing particle, you could create it enzymatically through ligation. And the mass action kinetics of ligation, or actually any enzymatic reaction, can effectively mimic Hebbian learning in neuroscience. So you might have all seen pictures like this in neuroscience talks, where these are different neurons, spiking patterns of different neurons. And people might draw a box like this to show coincidence spikes between neurons, right? These are neurons 1, 2, and 3. And the reason they might draw that and write an equation like this is to say neurons that fire together, wire together. They form a synaptic connection. If instead you interpret these three lines as the concentrations of different molecules, and imagine you had an enzyme, a ligase, that simply ligates together pieces together, these coincidences effectively mean that you'll create a new molecule IJ that is a glue between I and J at a rate proportional to the product of the concentrations, which is exactly the Hebbian rule of neuroscience, right? So there's not quite a complete closed proposal, but the idea is, in principle, you could learn gluing particles. You could learn interactions because you can create gluing particles based on some inputs, and then those could then mediate the interactions needed for a neural network like what I presented.
So Arvind, when you're training, you'd be challenging the system with glue fragments, and then when you're recovering, you'd be challenging it with the
building blocks. That's right. Yeah. That's right. You're changing. Yeah, I'm changing language here on you a little bit, but that's right.
Yeah,
that's right. Oh, I guess this is well, this is, I think, my last or second last slide. So I just want to summarize, saying big message. Why does matter? How come matter naturally mimics a neural network? It's because of two things. One, I had disorder in interactions, promiscuous disordered interactions. And two, you needed non-linearities. In this case, it was provided by nucleation. But besides that, I would argue this is a generic system, right, of promiscuous interactions with nucleation. I mean, you get nucleation for free when you have crystals. That's one theme. The other thing I introduced at the very end is if interactions are mediated by other particles, maybe you can someday learn these interactions. The final theme, which actually I really care about, but I didn't mention much, is sort of a lesson for biology, that in biology, in molecular biology, you usually assume that molecular promiscuity is a bad thing because things become uncontrolled. This is an example where there is a lot of molecular promiscuity, but the system as a whole does some complex specific computation. Like this input is mapped to that output, right? These concentration patterns result in that particular output. And so it's sort of the specificity through promiscuity effect. And this idea hasn't been explored that much in biology, with some really cool examples. One of which is this work from Michael Elowitz's lab by Yaron Antebi, who's now faculty at the Weizmann, in my view, a somewhat similar idea, but just in a real biological system without large nucleation effects, of essentially receptors and ligands in the BMP pathway that have extremely promiscuous interactions. So this is the pathway in mammalian cells where there are a whole host of receptors and many ligands, like let's say at the order of 10 and 12, and they don't interact in a one-to-one manner. They interact all over the place. Each ligand binds to multiple receptors with different levels of affinity and so on. But the net effect of that promiscuity is to effectively do a computation and have specific input-output responses. Right? So to me, that's another theme that's worth thinking about in biology and maybe testing in synthetic systems. Well, with that, I think I'm done. I want to, again, thank the people who actually did this work. And these are people, other people I work with in my group who working on other problems with somewhat similar themes. That I'll stop. Thanks, Tom.
Yeah. It's always weird because everyone's on mute, and therefore it's just like, you just get a deathly silence.
Yeah, I forgot to unmute in time.
Well, we had a lot of questions as the talk was going on, but yeah, I guess we open the floor for any more discussion. I mean, thanks very much. That was great. Really enjoyed it. I think the questions showed that people were interested too. Jonas, are you able to ask the question yourself, or do you want me to read it out? We've got. Oh, noisy back. Okay, so Jonas Hartman says, by analogy to tiling/tessellation-based computing, do you think such systems could be Turing complete? Question mark.
Yeah, I think these things are. I think the answer to that question is yes, but to me it's a different view on self-assembly. So in fact, Eric Winfrey, I think, made his name many years ago, proving exactly things like that. Right. So those approaches are more like the first neural network I showed you, where you mimic. Let's say you have an algorithm, and then you sort of compile it into molecules, element by element. I guess what we're going for here is more a sort of a statistical view. You just have a mess of molecules. You don't really design it to do something. I mean, you have these promiscuous interactions, but then just what kind of interesting computations or pattern recognition or sort of statistical computations can this mess of molecules do? Right? So it's anyway. The answer to your question is yes, but I would say the angle of this is towards a different kind of computation.
Arvind, I have another question. So first of all, thank you very much. It was very, very enjoyable talk. It was really brilliant. So you have shown the 2D structure of this. I was wondering, do you think this? I mean, could this be extended even to 3D structures?
Yeah, thank you. So I mean, theoretically, the answer is of course yes. You can extend it to 10D structures, and it gets better and better theoretically. And I feel like this is one aspect I didn't really manage to communicate, which is this flexibility question. Like, why did we pick these images of people and try this? The 2D structure really does restrict you in the kinds of pattern recognition problems you can solve.
Exactly.
Yeah. I mean, maybe it's intuitive enough. I'm not able. I find it somewhat intuitive, but I'm not enough to communicate it. But the answer is 2D structures can solve less complex problems than 3D, which can solve less complex structures than higher dimensional structures should they exist. But maybe your question is also technologically, is it possible? I wouldn't personally try it, but these people, but actually maybe one should, because I can go back to that slide. Peng Yin, who did these 2D structures, soon after did that in 3 dimensions. Like he created a whole host of 3D structures with the same simple logic, right? I mean, I presume in practice the experiments will be more difficult, but yeah. And in fact, one pet toy idea we've been throwing around is related to the last question as well. This whole thing I talked about was all about nucleation. Nucleation did the interesting stuff. The growth was kind of boring. It's whatever. It just grows out. Maybe it leads to depletion, but it was all about nucleation. The last question got at computation through growth, which is sort of the dominant idea in the field. And if you have a 3D structure, you can imagine exploiting each one in sequence. Like you have 2D faces, and each time a new 2D face nucleates, that solves this kind of pattern recognition problem. But then in its growth, maybe it executes one of these Turing-like computations, algorithms in its growth, which then decides, the nature of computation is a bit different, but 3D structures get to combine them.
Thank you.
Hi, Arvind.
Yeah, go ahead.
Oh, no, maybe you could go first. Oh.
Go ahead. I didn't have such a deep question. Maybe you have something more interesting.
Oh, I don't know, but okay, I'll try. Thank you, Arvind, as well for a really amazing talk. I really appreciated it. So I wanted to just pick up on the idea of whether you think this would be qualitatively different if you then tried this on proteins instead of DNA, because of I think one of the assumptions is that the interactions that you pre-build into your logic with the DNA are always the same, right? So, I mean, you always get the same number of interactions, is that right?
I mean, in this model and in these experiments, yes, but I think that almost doesn't matter, provided that the average coordination number matters. So like, which is the same as the dimensionality answer I just gave, right? So you want it to be coordinated enough, but certainly you can have a more like a ribosome-like interaction where some, bind to 5, some bind to 2. But anyway, go on. Yeah.
So, I mean, the point being that in the different states of assembly of, let's say, multi-protein systems, you might have a very different, let's say, free energy of the assembly, right? Because you have very specific interfaces that bind in one state, in one assembled state versus another assembled state. And so the question is whether that would really impact on this ability to learn the molecular interactions.
Right. I would guess the answer is it would completely ruin it if all assemblies were at equilibrium, because then those binding energies will dominate, nucleation won't matter. But even in that case, so I would describe that case as a problem where you have the nucleation effects this talk was about, plus equilibrium effects. And, if they were both present to some extent, at least some of the story should still hold. If it's all at equilibrium, none of this will hold. And I don't know. I mean, many macromolecular structures in the cell. I mean, I know of some cases where it is a kinetically controlled process, but yeah, it depends on how kinetic the process is, I would say.
Yeah. I mean, there's an analogous system which is kind of an inverse problem, I would say, which is not inverse in the way that you described it, but in the sense that rather than having a mixture of many different building blocks that then form a variety of different states, you could have the same building block, or you could pose the question of, does this building block self-assemble or not? And I wonder if that's approachable and in a learning context as well, because that's like a virus capsid assembly problem or something like that.
Yeah, that's interesting. I mean, right. I mean, I know of some work like this. You mean with a single building block, right?
Yeah.
Right. But then the only parameters you get to change are often, like, let's say the interaction potential as a function of R, like as a function of distance. And I know some people, like Sal Torquato has tried, actually many people have tried solving the inverse problem, Like, you want a 2D hexagonal lattice versus that lattice or a diamond structure. Yeah, but maybe you could also do it in a learning context. I don't know.
Yeah, I'd be interested to see how amenable that kind of problem would be to the protein case. I think it's really nice work. Thank you very much.
Well, thank you.
We had one more question in the chat from Ian Robinson. He said he'd like to ask a question.
Yes, thank you. Yes, I wasn't sure how crowded we were, but also many thanks for a very delightful talk, very thought-provoking. And I'm certainly not an expert in any of this, but I'm very curious. It's related to the question about dimensionality. I'm very curious why you're looking at 30 by 30 size systems here. And I'm wondering how it would scale if you went down to very small systems. I guess it would become rather trivial, but also in the other direction, if you go to 300 by 300, if all of these things are going to break down, because the number of nucleation events is sort of countable. It's of order 5 to 10. And I think something that may be actually important for the selectivity of the system.
Right. No, so I would say it's almost certainly going to break down if you go to smaller structures, because this whole thing, in some sense, the theory is all worked out to the large n limit. This thing works very well when you have a large number of components. When instead of 30 by 30, I think 300 by 300 would work much better, because, for example, even statistically, if you have 10 molecules that are at high concentrations and are localized on one structure, so 300 by 300, there's almost no chance, by random chance, that it's going to be localized to the other two. That, and also just, the theory of nucleation, the non-linearities in it, all are much stronger the larger the structure is. If you go to small structures, I would expect all this to break down, because the concept of nucleation itself, is not going to apply. Once you go to, I don't know, I'm going to say, I'm going to guess 5 by 5. If we tried the same thing with 5 by 5 things, it's probably not going to be a strong nonlinearity. Like the nucleation barrier might be like a 2 by 2 tile, and you can't really do much with that.
And that relates a little bit to the question of how many images you can classify, because it did look like it was the ratio of the number of nucleation events to the size of the object that was determining how many mappings you could make.
That's
and
right.
that will get better as you go to larger systems.
Exactly.
Yeah, thank you.